Well, thank you very much, all of you, for attending this session, and a special thanks to Petra. Hello, Petra. How are you? Thank you for coming. I will give a very short overview of your professional achievements. Petra Ritter is full professor and director of the brain simulation section at the Berlin Institute of Health, Charité University Medicine Berlin. One of the sentence that sometimes you have highlighted with respect to your work is that your vision is to plan the treatment of brain disorders using a digital copy of the person's brain. I don't know if this is very ambitious or not. This is something that we will learn very much about this morning, especially with respect to your most probably important tool, that is the virtual brain. The Petra Ritter studied medicine at the Charité University of Berlin. He has been working and making research internationally at the University of UCLA and UCSD in Los Angeles, the Mount Sinai School of Medicine in New York, and the Harvard Medicine School in Boston. In 2002, she receives her title as a medicine practitioner, and in 2004 she completed a doctoral thesis at the Charité, and she received in the specialty of habilitation in experimental neurology. After being Max Planck Minerva Research Group leader from 12 to 15, she assumed the lifelong position of Johanna Quadrant Professor Berlin Institute, one of Europe's largest university hospitals. And she's from 2017 the director of the brain simulation section at the Charité. The current research focus of Petra is brain simulation technology for personalized medicine, based on her previous work about neural oscillations in healthy and pathological brains, multimodal brain imaging with simultaneous EEG, fMRI, and brain state dependency of plasticity and learning. Well, that is, I know that is a very short mention to your prolific career, Petra. I wish that not many important things are missed in this short description. Thank you very much for coming to this active group that we have in Spain, that as you will learn, it's composed of different hospitals, clinicians, practitioners, also researchers in different universities, and also some industrial actors. So the floor is yours. Thank you for coming, and I would like be very happy to learn more about the virtual brain. Thank you, Petra.
Thank you very much, Guillermo, for this really kind introduction, and thank you for the invitation, for the opportunity to present. It's a pleasure. Also great to see so many attendees here. So thank you very much for taking the time. I am sharing my screen now, and I hope everything works well. Is it visible fully? I hope so. Can you please knock, Guillermo? Yes. So it's I did not hear anything. I hope it is visible. So
you're seeing it fine, Petra. It's
okay. Great.
See the title. Yeah.
Perfect. Thank you very much, Barbara. So I'm going to speak today about the virtual brain cloud, which is a relatively recent development as part also of the Human Brain Project and the eBrains platform. And the virtual brain cloud enables complex simulations of multi-level, multi-scale health data in a distributed cloud environment. Quickly about my background, I am, as mentioned already, heading the brain simulation section at the Berlin Institute of Health at the Charité University Medicine in Berlin. So this is the main patient building that you see here, and around this building there's quite a big campus, and there are actually 4 different campuses in Berlin distributed, where all the labs are. So my lab you can find in this building under the roof with the green grass. As we all know, the brain is complex, and that takes quite a lot to get some understanding of this complexity and of its function. And as you also know, since you're part of it, the Human Brain Project and the eBrains platform enable large-scale collaboration of hundreds, if not thousands, of scientists in a very effective manner in order to get a better understanding of this mystery of the brain. The Human Brain Project started in 2013, and interestingly, also in 2013, we published this article. Now I have to remove this little window from Zoom that I can read it. So this was a publication in the Brain Connectivity Journal. The virtual brain integrates computational modeling and multimodal neuroimaging. It was in 2013, and it followed the release of the brain simulation platform in 2012. Yeah, so the brain simulation platform is an open source platform that is available for free via the web, can be downloaded. These are the lead institutions: Marseille University, Baycrest, a hospital that is part of the University of Toronto, and the Charité. And in 2012, we also actually, even though it's open source, we put in a patent application, a provisional patent application for this new technology. Now, 10 years later, 2022, if you go to the website, you will see from the website itself, we had more than 40,000 downloads of the software. And just in the past 30 days, there were more than 500 additional downloads. So there's a maintained interest in the software. And the website is not the only place where the software can be downloaded. It's also available, for example, from Docker Hub. So this is not the full number of downloads of the software. We are very happy to see that also many publications were using this simulation framework. In the presentation today, I will focus on the latest innovations. Yeah? So we had 10 years of the virtual brain, and I know that you also got a presentation by Victor Gierse from Marseille about the virtual brain. So the focus today is what has happened in the past, let's say, 2 to 3 years, what is new. And so what can be done with these innovations? What are possible applications? The topics that I will cover are multiscale simulations, digital drugs, in silico deep brain stimulation, a knowledge base adapter and ontology to systematically integrate biological knowledge in the virtual brain. Then the embodied virtual brain, this is the virtual brain connected to the neurorobotics platform, and the cognitive virtual brain, where we, I think, as one of the first use cases, show that the virtual brain not only simulates activity and dynamics, but indeed also captures brain function and can simulate performance, and also captures inter-subject variability in cognitive performance. But before I go into these innovations, I would like to quickly explain how these brain network models are constructed. This is a publication from 2015 where we have built and made available open source pipelines to process imaging data, multimodal imaging data, to reconstruct, for example, the tractography, the long distance fibers in the brain, to perform the parcellation according to different parcellation schemes, to parcellate the brain in subcortical and cortical regions. Then the tractography fibers are being aggregated towards the structural connectome that connects the different brain regions, and which is the skeleton of a brain network model. And each of these brain regions then is equipped with these population models, mathematical equations that describe the average activity in a brain region. And now the appealing part of these models is that they can be simulated. So they are not just static models of the brain or atlases of the brain, but they generate meaningful dynamics, taking into account all the complex interactions between the different nodes. And this is what is being simulated. There's many different state variables. That is the firing rates of populations of neurons, the postsynaptic currents, inhibitory and excitatory currents, local field potentials, EEG and MEG signals can be simulated, taking into account the different layers between the neuronal sources and the sensors. And of course, then we can also apply stimulation and also deep brain stimulation, which I show you later with electrodes that are inserted in the brain, and record the observations. We can then add, of course, different features from atlases that we use for heterogeneous parametrization. That means that the different population models in different parts of the brain get different values for their parameters depending on the biological information that we want to integrate. Since the pipelines are digital and automated, and most of the pipelines that we have developed are also do not require manual interaction. There are some pipelines for EEG data that require a certain degree of manual interactions, but the MRI processing pipelines are fully automatic. It's feasible to process several brains at once. So for example, in this 2015 study, we had processed 49 data sets and generated 49 virtual brains. And now, 7 years later, the size of the cohorts has even further increased, and we are processing, for example, the UK Biobank data sets. So what is the benefit of brain simulation? One big benefit is that we get an understanding of the underlying principles that lead to the observations that we have in our experimental data. And here in this eLife study, I think it was published in 2018 or 19, we showed how simulating 22 minutes of resting state fMRI in 15 adult individuals also at the same time provides us with the underlying state variables for each brain regions, for example, the firing rates of the excitatory and inhibitory populations that you see here for each individual brain region. And we now can relate, for example, the alpha activities, this is a prominent 10 Hertz rhythm in the brain, to the ongoing excitation and inhibition firing rates, and to the slow resting state fMRI signals. And with this, it is possible to learn the principles of these relationships. So this is a little bit of fuller videos showing all the different components. And also here, not all components are actually shown. So in total, we linked 7 different empirical observations together in the model and identified the principles that link them together. So for example, on a very fast time scale, we reproduced the relationship between the firing of neurons and the 10 Hertz alpha rhythm. There's a relationship of the phase of such a 10 Hertz oscillations with the firing of neurons. And this relationship then propagates up in a relationship between the power of the alpha rhythm and the firing rate, between the EEG signal and the fMRI signal, which you see here, for example, on the top. And this translates to the resting state fluctuations that you see here. So we found the mechanism or candidate mechanism. After all, it's still a model, but the model is compatible with the mechanism of pulsed alpha rhythm related inhibition of the firing rates that then leads to an ongoing balance of excitation and inhibition that leads to a slow modulation of the local field potentials of synaptic activity and of the resting state dynamics in fMRI. And from our perspective, this is the degree of understanding that we want to reach, that we really know how are the different observations connected. And this is possible with brain network simulations because we have a biophysically grounded model. So there are two challenges, of course. This is all not easy. First, constructing such personalized brain network models is a complex endeavor, even if there's that nice simulation platform with a easy to operate graphical user interface. There's also a programmatic interface, but all the data need to be processed first and brought in the format with the pipelines. And then I mentioned the multi-scale co-simulation, so there are different possibilities what to do with the simulation framework. And all these different toolboxes related to personalized brain modeling are shown here in blue. They have been integrated with eBrain's distributed informatics infrastructure during SGA2, when we were leading the co-design project, the Virtual Brain. The goal was to make these simulation software packages directly available to the users. And what you see here in the middle are the core functionalities of eBrains. And here in this column, you see the different ways of deployment or possibilities of usage of the Virtual Brain software packages. So for example, here on top, the actual Virtual Brain simulation engine can be directly opened or operated from the eBrains portal. So if we go to ebrains.eu service, the Virtual Brain, we can click the application and then it will run in the eBrains cloud. It also uses an HPC backend. So if there are computationally demanding simulations, and often they are demanding, then via the RESTful API and the Unicore interface, automatically the jobs are transmitted to the HPC backend and will use the resources available there. It's also possible to use the Virtual Brain via other deployments, not via the graphical user interface, but for example, via Docker containers that can be directly pulled from eBrains Docker Hub, or via Jupyter Lab, where it is also already available as a software package, and so forth. And the same is true for the processing pipelines. There we have a Jupyter Notebook interface, so they can be run from a Jupyter Notebook. There's also a demonstration available. And also here, the actual processing takes place on the high-performance backend, but the operation can be done via the portal, via the cloud portal and the Jupyter Lab notebooks. And their interfaces to the digital atlases and to the neurorobotics platform, which I will show in a moment. So one challenge is the complexity, and we try to make it as simple as possible. The other challenge, of course, is it's personal data, and we are in the EU, where we have the general data protection regulations. And personalized brain modeling means processing health data, which is a special category of personal data that has a particular degree of protection. So in order to make the whole thing lawful, also as part of the co-design project Aid the Virtual Brain, we implemented protection mechanisms like access control, encryption, and sandboxing. So the data are always encrypted. It's a client-based key, so only the data controller, the lawful data controller, has a decryption key. And during the processing at the high-performance computer, of course, the data are being decrypted with the decryption key, but during that time, the data are in a sandbox that is created to protect the data. Not even administrators of the system have access to the data during the processing. And the access control is integrated with the eBrains access control. So a single login is sufficient for identification and authorization of the person. And this has been published in this recent publication, 2022, Brain Simulation as a Cloud, the Virtual Brain on eBrains. And there were 31 institutions, eBrains partners. You will see very many familiar names from eBrains involved in this integration work. So it was mentioned by Guillermo in the beginning that the goal is clinical application, and one innovation towards that goal was to integrate PET data, positron emission tomography data, in our brain network models to again increase the biological realism. Yeah. So here in this study we included beta amyloid, and from the literature we knew that in the vicinity of accumulation of this protein that is aggregating in particular in Alzheimer's disease patients, in the vicinity of these plaques, the beta amyloid plaques, the inhibitory neuron function is impaired, which leads to disinhibition of excitatory neurons, hyperexcitation, and excitotoxicity. And it is thought that it even leads to cell death because of this hyperexcitation. And this coupling can be integrated in our models. So now we have, from the PET data, the burden of beta amyloid in each individual region. We have a coupling transfer function that modulates the degree of inhibition towards the excitatory neurons. And we have the PET data from the Alzheimer's disease neuroimaging initiative, not only for Alzheimer's disease patients, but also for healthy controls and for patients with mild cognitive impairment. And we integrated this data for all groups, and only for the Alzheimer's disease patients, we found, after integration of the PET data and the implementation of the mechanisms that I just explained, less inhibition, basically, a slowing in the simulated EEG. Yeah, so you see here a theta activity compared to the more alpha-like activity that we find in the simulations for the healthy control. And this slowing is indeed found in patients with Alzheimer's disease. So with this, we have a candidate pathway how the beta amyloid leads to the slowing that can be observed in the patients. But of course, we also want to know what can we do for the patients. And what we did here, also as a proof of principle study, we now applied a virtual drug. And the virtual drug that we applied is an NMDA antagonist. The drug is called memantine and is really used for patients with dementia, including Alzheimer's dementia. And if we apply such an NMDA antagonist in our model to our Alzheimer's disease patients that have the slowing, the pathological slowing in our simulations, we see that the slowing effect can be reversed. This is a dotted line. So Alzheimer's disease plus memantine shows the behavior, the frequency behavior in the simulated EEG similar to the healthy control in the patients with mild cognitive impairment. If we now look at the effect of the NMDA antagonists in our brain model, we even can identify a potential protective mechanism. So what do we see here? We see on this axis the dose of memantine. Yeah, going in this direction, we have a higher dose. On that axis, we see the input that drives the pyramidal cells. And on that axis, we see the output from the pyramidal cells. Now, if the input to the pyramidal cells increases because there's less inhibition, then of course also the output of the pyramidal cells will be higher. And this leads to that hyperexcitation and excitotoxicity and cell death. But if we now increase the dose of memantine, the NMDA antagonist, we see that with increasing input in the pyramidal cells, there's actually a little buffer that takes away the immediate increase of the outputs and the hyperexcitation of the pyramidal cells. So it may be this little buffer that we see here in our analysis that acts protective for the patients. And indeed, interestingly, this is not just a theoretical analysis, but experimental data giving memantine NMDA antagonists to patients with Alzheimer's disease showed also a temporary decrease of the pathological theta activity in these patients. This was published in 2013 by Claudio Babiloni. So now I showed you previously that we can use the model as a mathematical microscope because it gives us the underlying state variables that we cannot measure easily. So we measure fMRI, we measure EEG, we measure PET non-invasively, but we cannot really stick electrodes in all regions of the brain and measure the firing rates and local field potentials. In some patients, some electrodes are put in the brain, but they only are very selective and do not reach all regions of the brain. But with the model, we generate the underlying firing rates and local field potentials and the excitatory and inhibitory postsynaptic currents of the biophysical model that underlie the observations. And we can use the features of those which are representing more closely the actual neuronal dynamics than, for example, fMRI or the EEG that is measured on the scalp, and increase the feature space in machine learning classification, for example, of different disease groups. And this is what we have also done in this proof of principle study. Here you see, again, this is the Alzheimer's disease neuroimaging initiative dataset with healthy controls, mild cognitive impairment in Alzheimer's dementia patients, the different colors. And here we have just shown 3 features. There were more included, but the features included the local field potentials, the frequency band filtered local field potentials of all simulations in all brain regions, including in the thalamus. And it turned out that in particular, but not exclusively, the alpha band local field potentials in thalamic regions were decisive to improve the machine learning based classification between Alzheimer's disease patients, mild cognitive impairment patients, and healthy controls. And this has been just recently published, just a couple of weeks ago, in Alzheimer's and Dementia Translational Research and Clinical Intervention. So the hope is, of course, this is just the classification between the 3 groups, MCI, Alzheimer's, and healthy control, but the hope is to, in the future, find biomarkers more closely related to the altered neuronal interactions that early on can already point towards a potential disease trajectory in Alzheimer's disease. So then, of course, we are not aiming for building brain models that are one-to-one reflecting the reality, but nevertheless, for certain diseases, it's important to incorporate some biological information to make the model more realistic and more useful for a certain application. And to make this possible more systematically and in a structured way, we recently developed a virtual brain knowledge base adapter that links to large databases that have been generated with literature search tools. Skyview is the one that is being developed by the Fraunhofer Institute, Sky, that has annotated more than 35 million of papers, scientific publications, and of course, with new publications being incrementally added to the existing database. And based on this annotated literature, pathway inventories have been generated and are available already that, for example, link changes of entities related to certain diseases, for example, Alzheimer's disease or psychosis or epilepsy. What we did is we further developed these available databases based on the annotated literature. We found the locations, anatomical locations with which these changes or cascades have been associated. And with this additional spatial information, we can now map altered pathways to our brain models and directly map the effect on the evolving dynamics. How is this linked? So how do we link the knowledge base to the brain model? For this, we had to develop another tool, the virtual brain ontology, that systematically describes the parameters and state variables and their biological or biophysical meaning. We have now probably 15 different population models with different state variables and different degrees of detail of the description of the biological processes. And we have annotated them with mesh terms. So this is mesh terms are also used, for example, in PubMed. It's an existing, frequently used ontology. And based on this annotation, now all the parameters and features on the virtual brain get a biological meaning and can be easily linked to the features of the knowledge bases. What we also integrated in this virtual brain knowledge base adapter is a link to the Zebra client, Python client that interfaces the multi-level human brain atlas and eBrains. So with this interface, we can now pull information about, for example, receptor densities or also genetic expression into our brain model. So the advantages, we have already standardized reference spaces that we also use with a virtual brain. And we can trust, therefore, the anatomical anchoring and simply put on top of our model these different pieces of information. And if we have then a transfer function that links the observation to the specific parameters of our local models, we can directly analyze the effect these features have on neuronal dynamics. All the visualizations, of course, are kind of appealing and also interesting for the broad public. Therefore, we have also developed, based on the virtual brain, some public tools for public outreach. For example, this one was developed for the traveling exhibition of the Human Brain Project that was kicked off at the Bloomfield Museum in Jerusalem. And there was a big touch screen installed in the middle of the exhibition room, where then, when I was visiting there, a school class was like crazy, zooming in and out the brain and clicking around and finding information about networks and their role, functional role here for emotions, for example. And this is available in many languages. We add from time to time a new language. I think now we have about probably 7 different languages. This is an older screencast with less languages here. The tool is also available online, and everyone can play around with it. So we have it also in Hebrew, Arabic, and the last language that we added was Spanish, because we had a Spanish intern who translated this all to Spanish. So it's online. It should be already including Spanish as well. Then we are also developing these mobile apps where wireless headsets that measure EEG signals can be connected to the apps and the mobile device. And we have nice visualizations of this activity, but also have some games with which where we can use this brain activity to control the content of the game. Another very recent development that I found quite interesting was for the Technical Museum in Berlin, that will also travel after 1 year to the Technical Museum in Frankfurt. They were approaching us, and they want to have an exhibition on the topic argument. And they wanted to know what happens in the brain during an argument. And they give us a list of concepts, which you find here. And with our knowledge base adapter, we can now, from the more than 35 million annotated publications, identify the networks related to the different concepts that are all related in a way to the topic argument, like conflict, controversy, debate, despair, envy, greed, and so forth. And this is not the final app. The exhibition starts in October. We are still further developing it. But it's kind of fun to, as a side product, to be able to extract now such knowledge from such a big pool of. I hear someone speaking. Maybe if you could mute, that would be great. And this is just another exhibition which I never saw in person because it was open during the pandemics, but there's such a big tower in the Humboldt Forum. where we project the brain.
¿Quieres que veamos?
Okay, then I come to the end now. I only quickly touch the multi-scale simulations that we have recently developed. The technical part is described in this publication, and a use case has been also recently published where we simulate some parts of the brain with spiking neuron networks. So the advantage of multi-scale co-simulation is that the virtual brain is coupled with other simulators. We have coupled it by now with NEST, with Anarchy, which is also spiking neuron simulator. And we are just in development for neuron and NetPine interface. This is ongoing work. So what can be done with such co-simulation? We used it for deep brain stimulation, because there we have to bridge the detail of the subcortical nodes, the basal ganglia that we want to stimulate, where it is really decisive also where exactly on a sub-millimeter scale the electrode is located. And this has impact then on the firing rates and other parts of the basal ganglia, the thalamus, and the cortical activity. I don't go into the details, but just want to show you this is a publication in experimental neurology, that here we now for the first time also monitors spike trains. So with a co-simulation of mean field models on most part of the brains, but detailed spiking neural networks on some selected parts of the brains, such as the thalamus and the basal ganglia, we get the spike trains in these selected microscopically detailed regions. And for some questions, for example, also for spike timing dependent plasticity studies, the timing of the spike trains is relevant. This is also a very recent development that is not published, where with Fabrice Morin, we linked the virtual brain to the neurorobotics simulator via Gazebo. And by doing so, it's really interesting. We can reproduce a very well-known and experimentally well-described phenomenon that happens if you have the task to move your fingers antiphase, and then like this, then you increase your frequency. At a certain frequency, you will get in phase again. And this is the result of the dynamic interactions and the complex network, and is being reproduced by the virtual brain, and then is made visible by directly connecting the motor and sensor areas to this simulated robotic hands and fingers. And then I think this is one of the last slides I show you. It's also not yet published, but this is really work I love, and therefore I wanted to share it with you. It's now addressing not dynamics, but cognitive function. And of course, this is also relevant for clinical mechanistic understanding, for example, of dementia. Here we looked first at the 650 virtual brains, healthy virtual brains of the Human Brain Project, young adult brains. And we plug in the prefrontal cortex and posteroparietal cortex circuits that have been shown previously to be able to simulate working memory and decision making. And these circuits are being stimulated with an external stimulus. And now I will not show you all the details, because I could now talk an hour about what we are doing here. I just want to give you a glimpse and then an idea of what is happening. So here you see, for example, the correct decisions, the integration time it takes to come to a decision. And here you see the phase space that changes depending on the input amplitude. And the input amplitude that you see here is related to the functional connectivity. So what we show is if the functional connectivity and the degree of synchronization or desynchronization in the brain changes, this has direct impact on the correctness of the decisions, and also the time it takes until coming to a decision. And we can now trace the neuronal populations, how they reach after a certain time in a tractor space, and how this is facilitated or not facilitated via the functional connectivity in the background. This can be analyzed now systematically. And so this is my last summary slide, how we translate the virtual brain simulations and clinical applications. There are three main pillars. Number one is understanding the mechanisms. Number two is using simulated features to augment the empirical features to improve diagnostics or disease trajectory prediction or disease progression modeling. And the third is the potential for virtual therapy. I showed you the virtual drug therapy and the in silico deep brain stimulation, where we have proof of principle, and now we need clinical trials to really show that there's a benefit for the patients indeed. And what I did not show you today is the work that we started at the beginning of this year when we got awarded the call for the Service for Sensitive Data. This will build on our work during SGA2, where we built GDPR compliant workflows for e-brains. And now the goal is with the Health Data Cloud, the Service for Sensitive Data for e-brains, to build a federated infrastructure from the scratch with data protection by design and by default that enables the processing of sensitive health data and, for example, of imaging data or brain models in a federated environment that includes several IT centers. Partially, these are hospital IT centers. Partially, these are the existing supercomputing centers who are partners in this work. And I would be very happy to talk about this in more detail at another occasion. And with this, I'm ending my presentation. I thank you for your attention. I'm, of course, very happy to take your questions. I stop sharing to better see you for this.
Thank you. Thank you very much, Petra. There is plenty of innovations there. Plenty of interesting developments. I was personally impressed with the mapping of in colors of the emotions in the brain. I was looking for admiration, but this is exactly the kind of reaction that I have from your work. So I didn't find that. It's a very, very interesting work. Thank you for sharing with us. Any questions? Anyone wants to ask something to Petra to clarify any of some part of her presentation?
I have a curiosity. So at the mechanistic level, if you have explored the link between activity and plasticity.
Yes, actually, we have an older study, I think it was published in 2015 from our group, where we integrated spike timing dependent plasticity in the virtual brain. And we actually had a microscopically detailed model and translated the microscopically detailed model to the large scale. So this is a very kind of old study, but what we did more recently was integrating indeed the plasticity rules directly in our large-scale brain network model. So, for example, when I showed you the eLife study, there we have plasticity already included. And the plasticity rule is called feedback inhibition control. At that time, further advanced this plasticity rule, but at that time, it was the mechanism that was invented by Gustavo Deco, feedback inhibition control that adapts to the degree of excitation. Why is that necessary? Because if we do not implement this plasticity rule, we come in a situation that the activity very frequently kind of explodes in the brain. So we need to have an adaptive mechanism to make the brain behave in a naturalistic and biologically plausible way.
Thank you.
Yes, Gonzalo.
Yeah, thank you, Petra, for the presentation. I have a more practical question about the use of this kind of tools in hospitals. I understand that there are many downloading of the software, but I understand that now with the use of in a cloud, is it probably easier to enter into the use of this kind of tools? Do you have any experience of transferring this kind of technology to hospitals for clinical use? Or what is the situation at this moment?
Yeah, I think at the moment the users are still researchers, yeah, and indeed it's much easier to simply click on a button in the eBrains portal, and then you directly have the software available. You don't need to download and install it, because even then we see it in the mailing list, problems occur, and already this is the first barrier to download the software and install it correctly. But the users presently indeed are researchers and maybe clinician researchers also, but not routine clinicians who use it for their patients. That would be the ultimate goal for the future. But for this, of course, we need to make the outputs of the simulator useful for the clinicians. And this needs a lot of more validation and work before this, the outputs of the simulations really can be directly used by the clinician. I think you all are aware of the virtual epileptic patient that is closest to this target, because there the clinical trial is already running in the French hospitals. And if there's a positive outcome and there's sufficient evidence that the virtual brain helps to plan the neurosurgical intervention, then I think this will be one of the first applications where a direct interface for clinicians would be useful.
Okay, thank you.
Any other question for Petra? I would like to know, Petra, a little bit more about the drug digital innovation that you mentioned. What is more or less in a context is the feedback or the reaction of big pharma, if you have already have the opportunity to interact with them for potential collaboration and so on?
Yeah, so we had some interactions, and there was an interest, for example, by Pfizer to have a clinical trial together related to Alzheimer's dementia. This is still in the planning, it's not yet realized. But what I found interesting, there are companies actually that have the label digital drug already. And what they are exploring are actually intervention possibilities that act on a molecular scale, but are not related to giving a drug that you swallow, for example. But for example, using visual stimulation, different kinds of stimuli that then in a very, so that is hoped. I think there's no real evidence at the moment, but they hope to target signaling cascades and pathways in a very controlled manner. And for those companies, this is highly interesting what we are developing, because on the one hand, we are able to extract from the enormous amount of literature these relevant and interesting signaling cascades and pathways. On the other hand, we are also able to simulate the effect on those cascades and pathways upon different kinds of interventions or stimulations. And there we are speaking to some companies, but I have to admit that we never really kind of focused on pushing that forward. So it's just a loose interaction, but no real active program at the moment.
Okay, thank you. Thank you very much. Yes, Gonzalo.
I'm sorry, I was muted. Let me put another question for the future. In the last several months, we were discussing about the science vision of the brain research in the context of the brains. And one of the key concepts on the table was the advances towards a digital twin of the brain. I understand we are very far from that, but here, even in your presentation, in some cases appears the possibility to have something similar to that for small parts of small components of the brain. But what do you feel about? At what extent we could move in this direction, and what could be the next steps for that? Maybe in one specific part of the brain, of the human brain, like the hippocampus, to put an example, or do you feel that it's necessary to consolidate more the basis of the technology to advance faster in this process?
Yeah, thanks for this question. So I personally do not have a problem with the term digital twin, because I consider it a model, and it can be very coarse, of course. So digital twin doesn't mean, again, that it's a one-to-one copy, but it can be a more abstract model. And this is what we have already. Yeah? So we see already digital twins, and I have my own also in the computer, and it even looks like me because I also integrated the face surface. So there are clearly digital twins of the brain existing. The question is how useful they are. And this then at the end is a matter of the research, yeah, that we do with this technology, and also how we further advance the technology to incorporate more biological realism where it is needed, and really being guided by the question, for which purpose do we need to increase the biological realism? Yeah, so just without a purpose, it probably doesn't make sense to blindly try to become more detailed and more detailed. But there needs to be a purpose behind. For example, understanding, as you said, the mechanism of memory consolidation in the hippocampus. There's certainly such a coarse model like the virtual brain would not help you, yeah, because you need more detail, you need plasticity. But that is interesting to have the interaction between the scales. So you can have the large scale brain network and see how it interacts with a more detailed hippocampus, and how the global activity maybe changes on certain matters, like I showed you for the functional connectivity, that changes the performance of decision making. Probably it also modulates in a way the memory consolidation that is triggered by the hippocampus. So I think with digital twins that we have already, we can do a lot of nice research, and at the end it depends on the cleverness and the quality of the individual researchers who participate in this initiative, how useful the research will be. So I'm absolutely in favor of that. And what I think, what the overarching benefit, of course, of eBrains and the Human Brain Project is indeed the collaboration, yeah, the exchange. We often are frustrated because there are so many meetings, so we are always pushed to exchange. But this is also the big advantage, that we really come together, that we learn about the approaches by the others, and that we try to integrate this and make it even more easy to integrate it by bringing this all together in a single platform.
Thank you, Petra. A lot of work to do.
Absolutely, we will not be jobless in the next years. That's good.
Some other question? I have another one. That is I have already asked you about the big pharma, but I would like now to know from you one or two messages for startups in pharma that are making very interesting things that could potentially be enhanced with the virtual brain. As you know, we are developing solution workshop with different startups in brain, neuroscience and neurotechnologies. Very recently, we have organized a workshop on neuropharma, and we have a collection of startups that are really doing impressive things and molecular design, for example, and so on. What are the one or two messages that you could give us related with this eventual utilization of the virtual brain for them, and also not only for using the tool, but also potential or future collaborations or engagement with the work you do? So that is my question.
Yeah, so I do not know exactly the backgrounds of these startups, but I think if they are interested in discovery of mechanisms, that can be targets of drugs, then what we are developing, and this is a resonance that we get, is extremely interesting. So especially the combination with the knowledge bases, because this is a systematic approach to all the existing knowledge at all different scales that is annotated by literature mining tools, that is developed further to pathway inventories that links the different entities via relations. And then the last step is linking these very complex cascades. And I think I showed you one picture showing these cascades. So these are extremely dense knowledge graphs that we are generating, not the type of knowledge graph we have in eBrains, but it's a knowledge graph related to the existing knowledge. Linking this, coupling this to powerful simulators really enables us to see the direct effect of an intervention on very complex cascades in the brain, for example. So my personal feeling is, and also the feedback I received from some of those startups or smaller enterprises, that this will be highly relevant. What we have not yet done is thinking about making this a business model. So what we do usually, we meet with these, we even do some searches, we tell them for free what we find, and they find it really interesting. But I think doing this in a systematic way could be really a business model. It would be very interesting to look from that perspective on the possibilities of the technology that we are developing here.
Okay, thank you. Thank you very much for your answers and insights. Okay, well, I think that we have arrived to the end of this interesting meeting. I would like to give you our thanks on behalf of the innovation team and all the members of this Spanish community. I guess that you or all people here are able to contact you through the email that we can, if you don't mind, we can share. And well, and thanks for everything, and see you soon, Petra.
Thank you very much, everyone. Have a nice day, and we will see each other soon, I assume.